So the clinical genetic testing sphere is increasingly large these days. I live very solidly in this diagnostic space where we have a patient with a series of phenotypes suggestive of a genetic disorder that is being sent for genetic testing to try and find an answer, either a unifying diagnosis that explains all of the phenotypes or maybe multiple diagnoses that together explain them. However, this clinical care continuum is increasingly expanding more towards the preventive side in genetic testing, where you may have some predictive tests for susceptibility for adult onset or even childhood onset conditions in the form of newborn screening, prenatal testing, or carrier testing to help with reproductive planning, et cetera. So in these different forms of genetic testing, while the content that you might include in these tests and the types of variants you would report back may change, the general process for classifying gene-disease relationships and variant pathogenicity should not change. So I'm going to spend a fair amount of time today talking through some honestly pretty onerous guidelines that the field has developed for classifying variant pathogenicity and classifying gene-disease relationships. And before I take you through those, I just want to give you a sense of why we go through this process, why we put ourselves through this. And it's really that the results of genetic testing, as with many other medical tests, does impact lives. So it's very critical that we get these interpretations right. Overinterpretation of these tests can lead to a patient, for example, getting diagnosed with a disease they may not have. This may then result in additional follow-up tests that have their own costs and risks associated with them. And it doesn't just apply to that one individual who got the genetic testing originally. They may then go and do cascade screening in their family members. So it can expand out to impact the entire family as well and impact reproductive decision-making. So the article at the back here is actually one from The Atlantic, where they're describing the story of a family who had some abnormalities reported in a fetus on ultrasound, ended up doing some prenatal genetic testing related to that, and received a likely pathogenic variant on their prenatal genetic testing report. The lab interpreted that variant to the standards of the time and to the best of their ability, given the evidence they had at the time. But it turned out that several years later, that variant, and the family decided to terminate the pregnancy based on that information. But it turned out that several years later, that variant was reclassified to a likely benign variant. So that's obviously an undesirable outcome. On the flip side, the underinterpretation of genetic testing results may lead to a patient not getting a diagnosis that they perhaps could have received based on the identification of a variant and the evidence available for that variant at the time. Obviously, this could lead to missed treatment opportunities, again, both for that one patient being tested as well as their family members. And we already have examples of lawsuits related to these kinds of mistakes. So the interpretation does matter, and that's why we spend a lot of time trying to standardize it. So in the world of a clinical geneticist who is trying to interpret the results of a genetic test, we are constantly considering two different axes of implication, and that being the variant level evidence, which we're depicting here on a y-axis, and the gene level evidence on an x-axis in this figure, which was still included in MacArthur. So in this top right quadrant, we have a very well-established pathogenic variant and a very well-established disease gene. So the example here is the Delta 508 variant in CFTR, the most common cause of cystic fibrosis, very well-established. Even in these well-established genes, we're going to encounter some variants for which we have a lot less information. So this would be a variant of uncertain significance, or a VUS, for example, in the CFTR gene. As we move back down along the x-axis there, you're also going to encounter, especially if you do broader scale genetic testing, some variants that we have little evidence for, and also genes that we have little evidence for. So this is a GUS, a GUS, or a gene of uncertain significance. And some people, boost and a boost. It's not much we can do with these. These really don't belong on genetic testing reports at this point until more evidence emerges. But what I want to especially point out in this figure is that this top left quadrant really cannot exist. You cannot have a variant that you are confident is pathogenic for a given disorder if the gene that that variant falls within or impacts is not in that disorder. So let's talk about how we decide whether a gene is implicated in a given disorder. To set the stage a little bit here, I want to give some framework of where we're at with gene discovery overall. So this is a nice figure from the Center for Mendelian Genomics, where these kind of blue-green hues are the subset of the protein coding genes within the human genome that we have some evidence have a role in a Mendelian condition, whereas the yellow and orange hues are those genes where we just don't have any known implication in a Mendelian condition just yet. So that's obviously the vast majority. But what's really interesting to see here, although we've made great progress in gene discovery, is that if you look at human constraint data and mouse model data, there is evidence that there's still another in the range of 10,000 Mendelian disease genes to be discovered. So as we're working through, particularly in broader like exome and genome testing, the types of variants we find, you're going to see a big spectrum of gene disease association evidence levels from the very well-established all the way down to the undiscovered. And even if we hone in just on those genes that have had some implication in human disease, there's a lot of variability, at least historically, in the field in how that information is integrated and classified ultimately. So as a readout of this, this is an admittedly a bit outdated chart of the hypertrophic cardiomyopathy panel testing available at 14 different US clinical labs. And hopefully what you can appreciate is that the number of genes that are included on that panel actually varies pretty widely between the labs, right? So you can see that a lot of labs are in the range of 20 genes, whereas others are way up over 100. There are several reasons for this, but one of the main reasons is just differences in how labs are deciding to integrate the information that's available in the literature to decide this gene has a worthy enough association with human disease to include it on their panel. So to begin to standardize the field in this respect, the Clingen group has put out these nice evidence-based framework for classifying gene disease validity. Before I go into that, we're going to talk a lot about Clingen today. So let me just introduce what Clingen is. This is a large NIH-funded project that has 3 major centers, one of which is led by Heidi Rehm here at the Broad. And really, their motive is to create an authoritative central resource that defines the clinical relevance of genes and variants for use in both clinical genetic testing as well as research. So Clingen has 4 major focus areas: gene disease validity, variant pathogenicity, dosage sensitivity, and clinical actionability. And in addition to doing a massive amount of curation work in these 4 categories, they are also committed to disseminating their knowledge broadly and freely to the community. So you will see many examples of really critical Clingen developed resources that we use in both gene and variant classification. So back to the gene disease validity guidelines for Clingen. Again, this is an evidence-based framework that considers both genetic evidence, the case level evidence, family segregation data, et cetera, as well as experimental evidence like your mouse models, et cetera. And I will say, though, this is the original paper on this framework published in 2017. Clingen has iterated on it a bit. And so you can actually find their current standard operating procedures for gene disease validity classifications on the Clingen website, which is linked at the bottom here. So one of the major contributions of this paper, in addition to the framework, was defining the terminology we use to describe the strength of gene disease relationships. Prior to these guidelines, every lab kind of had their own way of describing these, right? So one lab may have a red to green scale, where a red gene is one that is implicated in disease, a green gene is not. Other labs also use that red to green scale, but had the opposite scale so that a red was not implicated disease. So it was just hard to align on this when we weren't even speaking the same language. So this is really important to define these terms, all the way from definitive at the highest level of implication in disease down to no evidence reported, and even a few categories of conflicting evidence reported as well. It's a points-based framework. So you will see if you look at the top here that the genetic evidence can be assigned anywhere in the range of 0 to 12 points, the experimental evidence in the range of 0 to 6 points. And then in the middle, you can see after you add those points together, the final classification you end at. An important thing to note is that given that the experimental evidence is limited to 6 points total, you cannot get above a limited classification without at least some human data supporting this gene-disease relationship, which makes sense, I think, if you're considering a clinical implication of this. Another thing you might notice looking at these categories is that the strong and definitive categories actually have the same number of points. The distinction between these categories is really that the gene has stood the test of time, as they say, that there's been at least 3 years since the initial publication of the gene and no significant conflicting evidence has come up in the meantime. So in addition to standardizing how labs are interpreting the relationships between genes and diseases, this is very important for how labs are defining tests as well. So shortly after the publication of this framework, the American College of Medical Genetics and Genomics incorporated this into their technical standards for the design of diagnostic sequencing panels, recommending that sequencing panel tests should really only include genes in these definitive, strong, and moderate categories, and that limited evidence genes, for the most part, do not belong on these panels and should only really be addressed in more broad-scale sequencing like exome and genome. So back to our framework, let's walk through what it might look like to use this framework in the real world. So let's say you were, yeah.
And about those panels, given that genomics is changing every year, do these, under the hood, are they running exome or is it just gene panels?
Yeah, great question. So under the hood, the vast majority of labs at this point are at least on an exome backbone and increasingly even going to a genome backbone. And there are huge advantages to this, so that when you want to add genes as they are discovered, it's just a matter of bioinformatically unmasking them versus having to create a new panel with new capture. Yeah. All right, so we're doing an exome sequence. We're analyzing it for an adult with dilated cardiomyopathy, trying to find an answer for this patient. Say you found a variant in the ABCC9 gene, right? You may initially say, go to OMIM and see, well, you know, what is the role of this gene in disease? And you might be very encouraged to see, oh, look, there's a line for dominant dilated cardiomyopathy. Maybe this is very important for my patient. You might also do a little Googling and see that this gene is included on some diagnostic panels for dilated cardiomyopathy. So you do your due diligence and you say, you'll take this through the Clingen gene curation framework. And I know this text is a little hard, but I will walk you through the data. What you will find upon doing a literature search is actually that there is only really one paper that convincingly finds variants in this gene in a patient with isolated dilated cardiomyopathy. This paper from 2004, and there are actually 2 probands within that paper with DCM. One of them had a frame shift variant, so this was a loss of function variant, and in the framework, this would normally get you 1.5 points. But when you're looking around in NOMAD, you actually notice that there's a nearby frame shift variant in NOMAD, not the same one, but a nearby one that has a frequency that's a little high that you would expect for DCM. So you decide to downgrade those points a little bit to a total of 1 point there. You also find that there's a missense variant in a proband with DCM in that paper reported as well. That missense variant had some functional studies to suggest it impacted the protein, so that qualifies for 0.5 points. That's all that you found for your genetic evidence, but looking into some of the functional studies that are available, you find that there is expression in cardiac tissues, which gets you 0.5 points. you find that there is an animal model with some cardiomyopathy phenotype, as well as a rescue experiment done that rectifies that dilated cardiomyopathy upon reintroducing the wild type gene. So together, that is going to get you a 2.5 points. So overall, we have now a total of 4 points for this gene. This lands us very firmly in this limited domain. So ultimately, even though it looked promising at first, this variant cannot be interpreted as a diagnostic variant for your patient. It doesn't rule it out. Over time, maybe the more evidence will emerge for this gene. But given that the first paper was in 2004 and not much has emerged since then, hopes are not high at this point. So you can probably appreciate from going through this that this is not too bad of a process if you have only a few papers for these genes. That being said, it looked much easier just because we already had the paper summarized, and it takes some time to go and read through all those papers. But as you get to genes that have more and more evidence, a lot more papers to read, this can be a very cumbersome process. So it really does not make sense for every lab to individually be assessing all of these genes and duplicating efforts across labs. And so one of the major contributions of Clingen is to kind of centralize some of this curation work and also put it in the context of disease area experts, because interpreting what the functional studies really say about the gene can be complicated as well. So again, Clingen has established these expert panels that are disease area focused and go in and curate things like gene-disease relationships. And they've made great progress so far. This is a ton of work. And as of Tuesday this week, there's over 2,800 gene-disease curations available for free for everyone to use on the Clingen website. One thing that I think is particularly interesting to note, if you look at this little chart here, is that those red to gray hues in this top left section of the chart indicate that overall, Clingen is finding about 25% of the gene-disease relationships reported in the literature are limited or below evidence. So that's kind of a good warning for anyone who is thinking that maybe if this gene-disease relationship has been published, it must be real. It's definitely not true. A little more detail about what you might see if you went on to this information on the Clingen website. Again, it's all freely available there. You will note that genes can actually have been curated for more than one disease association, and that the strength of the evidence for each of those associations can differ. So for this particular gene, it's definitive for hypertrophic cardiomyopathy, only moderate for dilated cardiomyopathy, and then it has no known relationship for ARVC. There are also additional curation information on the Clingen website in addition to this gene disease validity. Again, we're going to touch on this a few times, but you can see information about dosage sensitivity curation, which is important for the interpretation of copy number variation that you might find in this gene, as well as the clinical actionability, which really reads into more of the types of tests that this gene should be included on. So Clingen, as I said, has done a lot of curation work. All of their information is available on their website, but they are not the only ones doing curation work in terms of gene-disease relationships. Every lab that is establishing a panel, for example, does a lot of pre-curation work to decide which genes should be included on that panel. And any lab that is doing exome or genome sequencing is also evaluating a lot of gene-disease relationships while they look through the results of that sequencing to see if it's relevant to your program of interest. You have a question?
Two questions. The first one was, what happens if you are hypothesizing that the variant is maybe impacting like a gene far away? Mm-hmm. How does that affect how you interpret disease gene validity? And the second question was, I guess if you find a variant in your patient, but it's not recorded anywhere else, but maybe there's like comparable variants in the literature nearby, how do you decide if if the variant that you have and the variant that's in literature, it's comparable, or it's like too distinct to like be weak evidence to use?
Both great questions. So the first one, remind me again, was
variants, I guess, in trans, like really far away from the gene.
Transacting variants. Yeah. So that is a great question. There isn't definitely specific guidelines on that. It would be really a matter of just looking, even if it's far away, that the gene that you think it's impacting has a role in disease as well, and whether there's other variation that supports that role, and whether you can prove your variant is impacting, for example, the expression of that gene, that would be sufficient.
Okay.
The second question, which is about other variants that may have a similar impact. We'll get to that when we do variant interpretation discussions, but there are specific codes for variants that impact the same position, for example, that are established as pathogenic. That counts as evidence towards the pathogenicity of your variants. We do also very much consider the variant spectrum of the genes. We'll touch on that a little bit as well, knowing that missense variants in this region, for example, are a hot spot. There's a specific code you can apply for that to blend pathogenicity for that.
Thank you.
All right. So as I was mentioning, there's a number of different groups doing this gene curation work. And previously, all of this information is just siloed inside those labs, right? They have their own databases that they're keeping, so they're not duplicating their own work, but it's not shared broadly. And so this was really the impetus for Heidi and Marina DeStefano, who's also based at the Broad here, to establish the Gene Curation Coalition, or the GenCC, which is a Clingen-supported public database of gene disease curations. The participating members at present are listed along the top here, and all of these groups are submitting their databases of gene disease curations to the website for everyone to see. This is an important resource because it kind of democratizes access to this information, but also allows us to see conflicts that are existing between these labs. There were always these conflicts. They existed before, but now we can see they're there and begin to address them, which is really important to improving the quality of interpretation across the field as a whole. So if you were to open up the GenCC website, which again is the gencc.org, you might see a page like this if you search for a specific gene, GJB3, and you can see every submitter's classification for any submitter that has submitted a classification for this gene, the strength level, and the disease-specific association, as well as inheritance pattern. You will see that the categories used on GenCC correspond to those that were recommended in the Clingen guidelines for gene disease interpretation, with a few extras. So, for example, this supportive category is not something you've seen in the Clingen guidelines, but it was designed to be able to incorporate some databases that actually are more curating the presence of a reported gene disease relationship rather than evaluating the strength of that evidence. We thought it was important to bring that type of information into GenCC as well. So everything submitted by Orphanet will have that supported tag, and soon everything from OMIM is going to be in there as well. And these are really valuable databases, particularly for bioinformatic pipelines, because it can allow you to annotate some of these gene disease relationships in broad scale. And again, the importance of identifying these conflicts, it allows us to go in and try to resolve them. So the GenCC steering committee calls, Marina will gather evidence on some of these conflicting genes from the available labs and present them together on the call. And then the steering committee actually goes through a voting process to decide overall what GenCC is going to kind of weigh in and decide what this gene disease classification should be. We don't get to the level of definitive or strong or moderate. We just decide, is it moderate or above? Essentially, should it be included on a panel test or not?
Do you ever incorporate like when the data was last modified or updated? Because I imagine, I'm just thinking that this is ever evolving.
In terms of the conflict resolution? Yeah.
Yes, and especially like on these displays, it's really helpful to know where they differ, but I wonder if where they say that there's limited evidence, maybe it's just old.
So in a different view on the GenCC website, you can see the submission date, which is very helpful as well, or at least the date that it was last assessed. And you can see that in Clingen as well. Actually, it's not to say that the expert panel classifications are right forever, right? They're going to change over time. So it is important to think about when that classification was done, whether there's new information that comes out. And we definitely do take in the age of that classification into account when we think about how we're going to go about resolving the conflicts. In particular, for the oldest classification, we'll often just go back to that lab with the older one and say, like, hey, have you looked at this more recently? And often they will say no. And when they do look at it, it kind of automatically resolves the conflict. Yeah. Okay. So we're like halfway through and we're just getting to variant interpretation. So let's move on. We can definitely talk more about gene curation at the end if we have more time, but let's make sure we're talking a little bit about variant classification as well. So variant classification, a lot of the themes you're going to hear here are very similar to what we talked about in gene curation. So variant classification, again, especially historically in the field, is subjective. Equally qualified professionals can interpret the same variant differently. And this can be because they may not have access to the same base of evidence. So for example, in my lab, I might have seen this variant de novo in a patient that was really convincing evidence for me, but another lab does not have access to that evidence. They may not interpret it differently. But even with the same evidence, labs can weight the evidence they find differently, which can arise in different classifications as well. So this issue has become particularly more apparent in recent decades for two primary reasons. One being that there's just a lot more labs doing sequencing, so more opportunity for conflict, and that the same labs, different labs are seeing the same variants. And that also there's this database ClinVar, which we're going to talk more about later. It's kind of the variant version of GenCC, which allows different labs to submit their classifications for variants. And that has just highlighted the fact that there are conflicts and that we can now see them. So if you look across ClinVar, in fact, about 23% of all the variants in ClinVar with multiple submitters have conflicting interpretations. So clearly there's, again, some standardization work to be done here. And I'm not going to dwell based on the time we have a lot in history, but this makes sense historically, just looking back and seeing what variant interpretation has been like as clinical genetic testing has been evolving. There were some guidelines for the interpretation and reporting of these sequence tests back in 2007, but they were very vague. It was kind of the wild, wild west back then. There wasn't really any standardized terminology about how to classify variants. And some of these guidelines are essentially, it was what was available at the time, but essentially it was like, you should check to make sure your variant is in another database, for example, or see if it's in another database before you go and report it. Didn't really tell you how to integrate that information and how to classify based on it. So luckily for us, a standard came out in 2015 that was a lot more prescriptive about what types of information you should look at when classifying a variant and how to integrate that information together. And this was a guideline developed together with the American College of Medical Genetics and the Association for Molecular Pathology. So in addition to telling us how to classify variants more or less, again, this guideline standardized the terminology in the field, which was an important step, suggesting that variants should be classified into these 5 categories that are listed in rainbow colors here, with pathogenic, meaning this is definitely a disease-causing variant, benign, meaning this is definitely not a disease-causing variant, likely pathogenic and likely benign on either side, which reflect about a 90% confidence level. And then everything in the middle goes into this bucket of what's called a variant of uncertain significance, or a VUS. Before, again, as with the gene curation, before the existence of these guidelines, there was a lot of variability in the clinical field about how the terminology that was used. So for example, a lot might say benign, or they might say polymorphism, or normal, or not associated, which is pretty confusing when you get that kind of information back on different reports. But these guidelines have really been very widely adopted in the field since they've been published. And as of 2017, about 95% of labs were now using these terms. So let's talk through a little bit about how these guidelines work. So if you were to open up this paper, you would see this nice, pretty rainbow chart, which is really the fundamental variant classification guidelines here. To walk you through it, I don't expect you to be able to read all the text in these cells, but just to walk you through it a bit, there's 2 major categories of evidence here, on the pathogenic side and on the benign side. In the columns, you will see that there are different strengths of evidence. So on the pathogenic side, the strengths of evidence range from supporting, moderate, strong, and very strong. And on the benign side, we have strong and supporting evidence. Now, in the rows, there are different classes of evidence that we consider in interpreting variants. For example, population frequency data, computational prediction tools, functional data, segregation within families, et cetera. So the general process here when you're classifying a variant is to go out into the literature, public databases, et cetera, pull all of the information you can possibly find about this variant, which is a task in and of itself. It's very time consuming. And then determine the pieces of evidence that you can find within those publications and databases and how they map to this grid. And then basically come up with a list of the criteria codes you might be able to apply. Once you have that list, you can go to this table at the right and determine how the different codes you have add up to a final classification. So let's walk through an example of what that might look like. Let's have this nonsense variant in the PAH gene. So PAH is an established causal gene for phenylketonuria, or PKU, an autosomal recessive condition. This variant is a loss of function variant. It's a nonsense variant expected to lead to nonsense-mediated decay, and it falls within a gene, PAH, in which we know that loss of function causes disease, causes phenylketonuria. So this allows us to apply a PVS1 criterion here. Just to give you a little overview of how the code naming works. If it starts with P, it's on the pathogenic side. It starts with B, it's on the benign side, and then the subsequent letters indicate the strength. So this is pathogenic, very strong criterion 1. You look in the NOMAD database for this variant to see what its population frequency is like, and you notice that it's in 1 out of almost 75,000 African chromosomes in NOMAD, which translates to a 0.001% frequency. This allows us to apply the PM2 criterion, which says either the variant is completely absent in NOMAD, or is at a very low frequency consistent with a recessive disorder, which is what we're looking at here. So in the 2015 guidelines, this allows us to apply the PM2 criterion. I'm caging that a little bit because there has been some evolution in thinking about this code, and we now only apply it at a supporting level, but we're going to go with what the 2015 criteria say for now and apply PM2.
Can I ask a question from online about that? Someone anonymously asked about how these pathogenicity encoding relates to genetic ancestry. And they're wondering, are there any examples of variants that are pathogenic in one ancestry but benign in another?
Yeah, so when we consider allele frequency, it does not have the ancestry within NOMAD does not have to match the patient being studied. And that's really because if this variant is at a high enough frequency in any population, it suggests that it cannot cause disease unless that disease is particularly enriched. So this is particularly true for continental populations. If you have a subpopulation like the Amish population, the Ashkenazi Jewish population, where that disease is enriched as well, you have to be careful interpreting your frequency cutoffs that you would apply. In terms of if there is any examples of variants that are pathogenic in one population and not in others, we actually do not assign variant pathogenicity in a patient-specific manner in that way. If a variant is pathogenic, it's pathogenic for everyone, but the overall interpretation of that variant in the context of your patient may change. There is maybe some option of background variation that may impact the expression of that condition or penetrance of that condition, and that exists, but we still call that variant maybe pathogenic. We may acknowledge on the report that it has reduced penetrance in some populations, for example. Okay, so then we do a literature search for the variant we're looking at, and we find that our variant is observed in a proband with classic PKU, who also carried a second variant on the opposite copy of their PKU gene, so another variant in trans. And that other variant, this missense variant at position 408, is a very well-established pathogenic variant for PKU. It's classified as pathogenic, for example, by 18 different labs in ClinVar. This allows us to apply the PM3 criterion, which states that for recessive disorder, this variant was detected in trans with a pathogenic variant. So In total, after doing all of this review, we find we have one PBS1, one very strong code, and 2 moderate codes applied. And if we go back to that chart on the last slide, you would see that this adds up to a pathogenic classification for this variant for autosomal recessive PKU. So this is kind of a nice, clean example for the purpose of the talk, but the more you think about these criterion and really work with them, you'll find that a lot of them are actually pretty vague, and it can be hard to decide when to assign each criterion, right? So that is because these classification criteria were really designed to be applicable across all Mendelian disease, right? So they had to be vague to apply broadly. But you may look in and you may see questions like, well, this code says a well-established functional study shows a deleterious effect. What does that mean? What's a well-established functional study? Or there's a code that says there's co-segregation with disease in multiple family members. So multiple affected family members carry this variant. But how many do I need to apply this code? And if I have a lot of affected family members, because I make it a stronger level, or the code saying that the minor allele frequency is too high for the disorder, how do you decide what is too high? So you can see there's more work to be done in terms of these codes. And again, this is work that Clingen has really taken on. So in addition to doing all of that curation work we talked about, the Clingen expert panels also work to further specify these codes to make them applicable to specific disease areas in a more logical way. So there are now over 30 disease areas that Clingen expert panels have designed these specifications for. They are available in this database called CSPEC, the link I've shown here. And this is a database searchable by either the gene, the disease, or the expert panel. And what you might see if you open up one of these, for example, is further specification of how to apply the codes in a disease-specific fashion. So this is an example from the MYH7 specifications, where you can see that if you look in the 2015 guidelines, this VA1 criterion just states the allele frequency is more than 5% in the databases available at the time. But for most disorders, 5% cutoff is more than an order of magnitude higher than necessary for most Mendelian conditions. So for this MYH7 criteria, they were actually able to specify based on the frequency of dominant cardiomyopathy, its prevalence, and the genetic and allelic heterogeneity of that disorder, that anything above a 0.1% allele frequency in databases like NOMAD is actually sufficient to classify that variant as benign. So Clingen has a group called the Sequence Variant Interpretation Group that actually oversees all of these expert panels in specifying the criteria appropriately. But this group also has published a lot of guidance on, more broad guidance on the use of specific codes, for example, the PBS1 criterion. What types of loss of functions qualify for that? What kind of caveats are there to deciding something as loss of function, and how you might up or downgrade that criterion based on the type of variant you're seeing. So all of that information is here on the SVI website that you can access. And having spent all of this time telling you about the 2015 guideline criterion, I will tell you that the SVI is now working on the next iteration of sequence variant interpretation, which is going to change things up a bit. So don't try to memorize the PBS1, PM2, because all of those code names are going away in this next iteration, still in development. So this is definitely not final, but the codes are going to be renamed based on the type of evidence that they reflect. And it's going to be a points-based system in the future, which is great actually for us in terms of the 2015 guidelines. It's very difficult if you have conflicting pieces of information, both pathogenic and benign, to decide how to integrate that together. But in this points-based system, you will have some positive points, some negative points on the benign side, and you can just add those together to end up at a final classification. Given that we spent time talking about GenCC, and I've mentioned a ClinVar a few times, I just want to highlight a little more that ClinVar is a publicly accessible database where many groups are submitting their variant classification information. The Clingen expert panels submit all of their classifications in ClinVar, but also the most variants within ClinVar are submitted by clinical labs, about 96% at this point. And if you haven't looked in ClinVar before, this is what you might see on a ClinVar variant page. At the top, you get an aggregate classification of what every lab has submitted. So for example, for this variant, you can see the individual submissions at the bottom. There are 3 labs that are calling this variant likely benign, 1 lab calling it a variant of uncertain significance. And again, as with GenCC, the existence of all of this information with one database has allowed some level of conflict resolution to go on over time. There is a star status. So one of the biggest problems with ClinVar is that there's not really a quality standard that has to be reached to get your information into ClinVar. So there's very widely varying quality of interpretations within ClinVar, but they've tried to help us to delineate some of that in this star status. So if a submitter has 0 stars, that means that submitter has actually not submitted their classification criteria that they use to classify the variants. So it's hard to know what they're integrating. Once you get up to 1 star, you've either submitted your classification criteria, or this happens also at the aggregate level when you have multiple submitters that are disagreeing. And then as concordance is reached, the star status will go up. So a 2-star variant has multiple labs saying the same classification. The expert panels get an extra star because they've really gone through this rigorous process, and we believe those classifications a little bit more. This is an extremely valuable database in terms of bioinformatic processing of data in clinical labs. So all labs at this point virtually will be annotating their data with ClinVar classifications as a means of pulling out previously classified pathogenic variants. There are problems with that? Hear the question?
The previous slide, after reading several ClinVar, and it seems like every lab adopted the guideline, but what was the hesitancy why they don't put in the codes in these reports? Is it that they don't want to be bound? But if they don't want to be bound, it's sort of hard for people who read to know what exactly is going on.
Yeah, I agree. So there are many answers to that. So labs overall are encouraged to submit their evidence, and you can see an example of an evidence summary here in that uncertain significance classification for this variant. But it is not required for labs to submit the evidence they've used to classify that variant. The reason that labs don't do it, honestly, is that they often don't have that information structured in a way to easily be able to submit it on large scale to ClinVar. It's a lot of work to get that information in. And so for the most part, giving labs the benefit of the doubt, I think that is the biggest barrier to getting this information into ClinVar. A lot of labs will have a contact button, or there's emails for every submitter in ClinVar. So you can go in and actually email that lab directly and say, what kind of evidence do you have for this? I do this fairly frequently for like Invitae and GeneDx, which are massive labs, have a lot of data in there. And they will often respond and give you kind of a summary of we've seen this variant de novo and ex proband. So when you have a very rare variant, you have no other means of getting that information, that's a good resource to try. But that being said, again, like it takes labs time to respond to these kind of emails. So it's not, massively scalable. It's a difficult problem.
Are labs incentivized to submit because it's a lot of work to do that? So are there any incentives?
Great question. Yes. So we have found over time that labs submitting to ClinVar is a great indication of the quality of their interpretation, particularly because you can see when they're like just way off on the vast majority of their classifications. So Clingen has actually developed, they have a site where you can see the labs that have they have a classification system essentially for labs that you get a higher quality rating when you've submitted a percentage of your variants to ClinVar, and you update it every year so you can see updates over time. So there's this lab list that is developed as the incentive for labs to do that. How often people who order tests go and look at that lab list to decide where to order from, I can't say. Heidi probably has some data on that. So as I was saying, ClinVar is a great source of annotation, but there are some problems with doing that. And that, again, as I mentioned, there's a lot of varying quality of data in ClinVar. So for example, if I filter a genome for everything that's pathogenic in ClinVar, you're going to get tons of hits, many of which are way too frequent in the general population to be pathogenic. Some of those were submitted before Exact or Nomad were available, right? So we really kind of need a way to clean up the database to use it systematically in this way. And that has been led by Clingen, which is now, Heidi has spearheaded this Clingen Curation of ClinVar project or CBC, in which Clingen curators now go in and review these conflicting entries in ClinVar, pick out, for example, the old ones that are outliers and flag them so that they're no longer integrated into that aggregate classification that's at the top of the page. So this is something that can be crowdsourced. So if you find errors in ClinVar, there's a link on the Clingen website where you can go and kind of flag them for review as well. So I just want to mention very briefly, we talked about the evolution of the sequence variant interpretation guidelines. There are also guidelines for the interpretation of copy number variation. In 2020, this paper came out, led by ACMG and Clingen. And this is, again, a points-based system for classification that takes into account genomic content, overlap with established haplosufficient and triplosensitive regions, number of protein coding genes, et cetera. Fortunately, I don't have time to go through that, but Clingen has a great web series that walks through these criteria and how to apply them. And I've linked that at the bottom of this slide. So overall, hopefully the take-home message is that the design and interpretation of genetic tests really requires a lot of detailed evaluation of genes and variants, and that the standards and guidelines that have been established, even though they are time-consuming, greatly improve classification, consistency, and quality in the field. I've listed out some of the guidelines that I've talked about, as well as a couple extras that I didn't get to. And then finally, that these public databases like ClinVar and GenCC are really critical to the field, creating transparency and crowdsourcing the work. So thank you to you all for listening. A lot of my slides were stolen from Stephen and Heidi, so thank you to them for putting these together. And a huge thank you to everyone that's involved in these Clingen working groups and doing really the laborious work of curation.